Great. Thank you for the students for organizing this. And my name is Andrea Stavoe. My first name rhymes with mitochondria, in case that helps. And I am an assistant professor in the Department of Neurobiology and Anatomy. And I started in September, so it's a brand new lab that we have going, all new equipment, which is really exciting. And so, next please. In our lab, we're interested in really broadly understanding how neurons are able to maintain their function during the lifetime of the animal in which they're in. Neurons are very unique cells with several properties that make it critical that they survive as long as the organism that they're in, but also make that exceptionally challenging. And so to understand how neurons are maintaining their homeostasis, we're studying a process in the cells called autophagy or autophagy. It's a degradative process in which the cell engulfs part of itself, either cytoplasmic contents or damaged organelles or damaged proteins, and sequesters those contents into an autophagosome, which then fuses with lysosomes and then follows the lysosomal degradative pathway to recycle those contents into their respective building block biomolecules. And so autophagy is especially important in neurons because they do have all these additional challenges. They're long-lived, they're highly polar and highly metabolic. And so they use autophagy to be able to maintain their homeostasis. And a great indication of how important autophagy is for neurons is that autophagy has been implicated in all of the major age-related neurodegenerative diseases. Myself and others have worked to understand how autophagy is spatially regulated and temporally regulated in neurons. And so we understand now that autophagosomes generally form in the distal end of the axon and then are transported back to the cell body where their contents are degraded. And so really broadly, we want to be able to understand how autophagy is regulated in neurons and all the different steps of the autophagy pathway are regulated so that we can work to identify therapeutic targets for these neurodegenerative diseases. And so we have a few broad questions in the lab. We want to understand how autophagy and all of its different steps are changing with age in neurons. We also are really interested in, once we understand how it's regulated, if we can ectopically modulate autophagy in neurons so that we can identify some therapeutic targets for neurodegenerative diseases specifically, but also so we can just generally extend the nervous system health span, both in terms of aging and in the context of disease. And so to do this, we use live cell multicolor spinning disk confocal microscopy, looking at different components of the autophagy pathway and different organelles in neurons grown in vitro. And we use these neurons from transgenic mice that we use in the lab that use GFP-LC3, which is a marker for autophagosomes. And finally, we also use C. elegans. The worm is a great model for aging. An old worm is only 3 weeks old. And they're also really great for in vivo whole animal spinning disk live animal microscopy. And we can look at individual neurons in these worms and specific components of the different pathways. So thanks for listening, and I'm happy to take any questions.